So as I've told you, this is an AI-based cancer classification problem. So in ML, that is machine learning, and DL, that is deep learning, and AI, that is artificial intelligence. So these are, ML is like machine learning. You, with the help of coding and some mathematics, you help machine to learn something. Now deep learning is much more deeper. You go in deep learning. What you do, you form layers. Okay, so you form layers, and the machine learns in different layers. So in first layer it will learn some 10%, another 10%. For that you have to make second layer. Okay, and then with the help of machine learning and deep learning, and if the machine is able to think like humans, that is artificial intelligence. So reaching towards a perfect AI is very difficult, but yeah, ML is now basic. You should know what is ML. Okay, and if you're very afraid that you do not you're not able to understand this project, don't worry. There are many videos available for ML. Now machine learning is nothing but it is based on mathematics, and that too you should know some basic of statistics. So as the class progresses, I'll just give you some videos to go through. Either it would be my video or some videos which I had learned from, so I can give you those videos. Nothing like, even if you know 10th to 12th level of statistics, it will be easier for you to understand what is happening. That's it. Fine. So nothing but maths and statistics. So before moving on, let me first um, tell you something. Can you see this word problem over here, or can you just are you just seeing the website still? Here there is a word problem, a mathematical problem. Can you is it visible for you?
Yes, it's visible.
Okay, so this is a question. Now why I have chose this is a ninth standard question of mathematics, and if you understand this maths, you will be understanding what our convolutional neural network model will do to predict what type of tumor it is just from images. How is it possible? Now this is a 9 standard second chapter problem from, and the it is a linear algebra problem. So you can see math, linear algebra. If you can understand this linear algebra, that is being applied in our, machine learning or deep learning or application of artificial intelligence. So that is why we are focusing on math also. But it's just like, you don't worry, you don't have to go deep. Now, this is a question. There's a rally which is starting, and in that rally, 120 members are participating. So what do we have? We have 120 members in the beginning. And each hour, 9 members are dropping out. Fine. So on the first hour, you have got 9 members less. And Afrin, can you please mute yourself? Dear, please. Thank you. And in the second hour, more 9 members will be dropping. So now a total of 18 members have dropped. In the third hour, 27 member would have dropped out. So can you tell me in the fourth hour how many members would have dropped out of 120? Can anyone tell me?
Minus 30.
Sorry, I guess you're not clear. What did you say?
Minus 36.
Yeah, how did you come to know this? I did not give you this data that on fourth hour, 36 people would be dropping out. So I'll be having, in the fourth hour, I'll be having like it's 11. So 84 members I'll be having, right? So how did you come to know? Is it right? It is right. You're right. Okay, but how did you come to know that 36 members are dropping?
Finding the pattern.
Very nice. And may I know who is talking? I'm sorry, but can you tell me your
Hariom.
name?
I'm Hariom.
Thank you, Hariom. Very
Hariom.
correct. So you found a pattern. Yes or no? What is the pattern? I will write it down. You already have 120 members, and in that, people are dropping out. So you have subtracted 9n. So this is a linear equation. Okay, this is a linear equation through which you have found a pattern. So your brain, other people also, if you have not understood that this equation is working over here, still you found the pattern without knowing the underlying mathematics. You found the pattern, right? Yes, we knew the table of 9. So something multiples of 9s we can look over here. Yes, and we knew in the fourth hour 36 people will be dropping, and in the fifth hour we'll have 45 people less from 120. Correct? So you have found a pattern with the help of mathematics. So you don't know this equation, or you don't know this expression, but you have found a pattern. But in case of CNN, what we'll do? So in case of CNN, first we tell them, as in we are going to tell the model, we will give them some equation or expression. We'll give some equation, or we'll give them some relation, and then it will find the pattern. So with the help of mathematics, it will find the pattern. What you did here, you understood the pattern first. You may be a small child, or the one who doesn't have a math in 10 plus 2, or the concept of linear algebra is not that clear. You might not understand how, within seconds, you couldn't understand how you got this expression. But anyhow, your brain understood that there's a pattern of multiples of 9. Likewise, first what we will do is we will give some mathematical pattern to the computer. You understand? And then with those mathematical patterns, with those mathematical expression, there will be some patterns. Okay? And with that patterns, it will look at the images and it will see whether the image is normal or the image is from tumor sample. So I hope you got some basic idea, everyone. Okay.
Yes,
Fine.
Yes.
So there's no need to be afraid. How will I be able to do this? Okay. So many people are jumping into, at least you'll have some knowledge of ML, some knowledge of AI, what is happening. So let's move on further. Yeah. So what you will understand, I hope it's visible clearly. Everything is visible clearly. I mean,
Yes, ma'am.
every one of you, are you online from laptop or are you watching this from phone? And if you are watching from phone, I hope the images are, I mean, the text are visible. I mean, not too small.
Yes, ma'am, it's visible.
Fine. Thank you.
Visible.
So, why histopathology based cancer classification? Now, what is histopathology? I will just tell you in second day, in day 2, that is on tomorrow. Okay. Why histopathology based cancer classification is an important and unsolved problem. This is a unsolved problem. Okay. Now, cancer classification, by looking at this image, we will classify whether it is normal or not, whether it is a cancerous or not. Okay, or tumor image or not. But our project is sub classification. Okay, what do you mean by sub classification? If it is tumor, which type of tumor? Okay, so even suppose we are having breast cancer data. So breast cancer is of multiple types, and we will look at our data set on the 7th or 5th day, I guess 5th or 7th day. We will have a class on our data set overview. In that we'll look ovarian cancer, how many types of cancer or tumor types you have, or in breast cancer, how many different types of tumor you have. So we'll look at that. So today is just an overview. Okay. Now, convolutional neural networks. So whenever you see NN or neural networks, now this neural network. So neural networks had come from the word neurons. Everyone knows what is a neuron. It is a nerve cell. And what is a nerve cell? So nerve cell has so many connections. Okay. Okay. So here, nerve cell is nothing but like a circuit. So many connections. And each connection is for learning something. So neural network is nothing. We will make this neural networks in the form of codes. We'll code in Python. Okay. And we'll make some nodes. Each node will be connected to other nodes. So we'll make some connections. This connection will be in the form of layers. So in neural network or deep learning, we always work in layers. So layer 1, layer 2, layer 3, we can construct how many layers we want, right? So in layer 1 it will learn first pattern, maybe like edge. In layer 2 it will learn other pattern like color. In layer 3 it will learn what is the shape. So like that, different layers it will learn. Okay. And neural networks try to mimic how the brain is functioning, how the brain is learning. That is what neural network is. Okay. You just grasp the concept for now. Okay. And we will move on slowly. Now, how we will also see how we'll build our pipeline or our project in 100 days. Okay. So what tools we are using, what are the data set. Okay. And beyond this classroom, beyond this our interaction, how the project will matter, right? So cancer studies are very a hot topic nowadays, and it will be in upcoming future, because if you think you have cure for one type of cancer, the other type of cancer is still remaining to understand. Okay. So the first, let us see what is the actual problem, real world problem, the clinical problem. It is the leading cause of mortality. That means it is the leading cause of death. And we have chosen breast and ovarian cancer. It is women related cancer. Okay. Now these two cancers, you can read most of it by yourself, but I'm telling you the important points. Now breast cancer can be detected soon. Okay, because there might be a lump in the breast or pain in the breast. Okay, or some kind of uncomfortable the women will undergo, and then she'll go for checkup. So breast cancer within early stages you can diagnose. Okay, early stage detection is possible. But when it comes to ovarian cancer, because it's so deep inside, and you cannot feel what is happening in ovaries, so usually the diagnosis is very late here, right? So these are the two contrasting features of breast and ovarian cancer. Now also these are the most commonly occurring diseases. Okay. And what we are using histopathological images. Other images we have, like we have X-ray images, we can have MRI images also. These all are medical images. But what we are using histopathology for now, you just understand, under the microscope, we are staining the cells or tissues and we are looking at it. We are looking at the morphology. Can you see the biology over here? We are just looking at the morphology of the cells. How the morphology is different for the tumor, and if all the cells are tumor cells or all the tissues are tumor tissues, how different type of tumor cells differ, what is the pattern of different types of tumor. Okay. Now in real world, the doctor must be very highly expert to select that pattern of tumor, and it is a time consuming process. It is subjective, as in maybe one doctor will identify a pattern as benign tumor. Okay, it's benign, it's not uh, uh, so dangerous, but other doctor may say no, it is malignant. So it's very subjective, it depends on how you perceive that pattern. Okay. Now medical imaging data is also okay. So these are something that why we are going to medical imaging data, but you can understand till here what I have told you, benign and malignant. Okay, so what one doctor is predicting, my other doctor might not agree with him, right? So usually the manual analysis will fall short. Why? Because you have to depend on certain person who is an expert. Then inter-observer variability. What one doctor is observing, the other doctor might miss that. And it is a time intensive process. One slide may take 30 to 60 minutes, or one case may take about 1 hour. Okay? And increasing data volume. Modern healthcare generate imaging data faster than it can. So this is like other problem. It's like computational problem, but till these three are the medical problem, right? So we need fast and accurate results. So that is why, rather than focusing only on manual, I'm not so whenever you build a computer model, don't think that manual analysis will be totally gone. Never. You should never underestimate the power of human analysis. You should always have an expert with you. Now these models are only and only to uh, lessen the burden of the doctor or the observer. Understand? So we are trying to make an automated, scalable, reliable diagnostic support system. That is it. None of the AI till now is meant to totally replace a skilled person, especially in healthcare. Okay. Now, a doctor, we have seen a doctor might look at this image, and there are many drawbacks. It's time consuming, it's subjective. So how AI will be addressing it? What it will do, it will take a tissue image. Now it is a stain slide. Now every biology student, okay, wet lab student, you know that we stain our cells with different type of dyes, right? So we have different dyes like saffronin, methylene blue, okay, malachite green. These are the stain that we are using, HNE stain. We'll just eosin stain something. So we will look at it in detail in the second day of our video, sorry, of our class. So what is HNE stain? Now stain is very important because it will give certain colors, and computer should understand what the intensity and what the different value of the colors mean. So color is very important. Microscope will capture that image. Now first you have to convert this image into digital data. I'll tell you in the second day how it is done. Okay, this digital data is then fed into the CNN model. After this CNN, that is convolutional neural network model, and then the patterns are extracted. There are some filters, and they will be using maths, they'll be using some expressions and some equations, and then you will predict whether what type of cancer subtype it is. See, predicting from normal to benign or normal to tumor is considered very normal project, but we are hopping just a step further. We know it is a tumor sample. We will just classify what type of tumor is it. So it is much more difficult than just binary classification. Binary means two level classification. We are not doing this. We are doing multi level classification. You'll all come to know about this. Okay. So I'll just give you another link after 10 minutes. When this class is over, please join again. Okay. Please don't go anywhere. Just a break of after this session is over. Just take a break of 5 minutes and come back. And I'll give you the link. Check your mail and check your WhatsApp also. Okay. Till then. Now what will this project will build? So we'll build a convolutional neural network and we'll classify again and again. So what we'll have a data set of breast cancer, and we'll also have we'll also have a cancer data set of ovarian cancer, right? So breast cancer is more complicated because when I started this project, I thought we'll only go for one type of cancer, but that is not that does not tell whether it is this model will be working on other type of cancer or not. So I've got time, so I use another cancer data set also, ovarian cancer. Maybe when we work together, we can build a model that we can feed any type of cancer and it will detect what type of, these are the major types. Okay, breast cancer, ovarian cancer, okay. But under breast cancer we also have 8 subtypes. In ovarian cancer we have 5 subtypes. So we hope that apart from the major classification, it can also classify subtypes of different cancer. Okay, now these are something: performance metrics, accuracy, precision, ROC, AOC. These are some technical stuff will we have to learn later on. Okay, these things I'll not go. So I think these things are already discussed if you have gone through the initial orientation video. So where this is used? So the domain is known as computational pathology, or it is also known as medical imaging analysis, or AI assisted diagnostics. So when we want to publish, we have to search these research domains. When you publish your work, you don't go for any journals. You have to um, stick to these domains. Okay, only then we have chance of acceptance of the paper. Then yeah, the technical stack. So we'll be using Python programming language. Inside Python, when we are working with deep learning models, so we have some packages. So these packages are used. So TensorFlow and Keras, these packages will come to know. So we have to download this one. Okay, we have to download these packages. Then while coding, every time we have to import also. So whenever we are dealing with packages in coding, after you have installed, each time you're shutting your computer and you're starting it again, you have to import at every run time. At every time you're running your code, you have to import those package. Only then those package will look at each line and they will work whenever they are called. Okay, whenever a certain package is called, it will work, but you have to import them. I hope a simple idea you have got. So the packages that we are using is TensorFlow and Keras. These are deep learning packages in Python programming. Inside that we have, okay, people who don't have high performance laptop, actually even if you have a high performance laptop, will not be working in your laptop because maybe your GPUs will be fried up or something. So we'll better use either Google Colab or Kaggle. Okay, they provide their own innate GPU, graphical processing units. So we need GPUs for deep learning, right? And at last we will be documenting it. Every work will be documenting, and you will have your own GitHub is a dumping ground of your code. What you have done, you have a readme file, a section which explains your code. You have different folders, you have your codes written over here, and you make your GitHub. And whenever you go, your recruiter will look at your GitHub account surely. Okay, so it is necessary to have, apart from resume, you must have the proof of what you have worked. So that is your proof. This GitHub account will be your proof, and we will look at how to set up the GitHub account. Okay, so by the end of this course, what you will be able to do, end of this project, you'll have a portfolio ready, reproducible deep learning pipeline. So reproducible is very important. Remember this, I'll tell you later. It is a deep learning, it is not machine learning, it is one step further, deep learning pipeline. Okay, so you will translate a biological problem into computational task. People take it very lightly. You think computational task is hard? No. We are very overconfident as biologists that we understand the biology. No. Once you go deep into any omics problem, any biological problem, if you know biology, this is only 10% of task, because we have vibe coding now and we can generate code. After the code is generated, then you can understand the code also. But if you don't know the biology, how you will be able to make the computer understand that biology? You should understand the biology first. Okay, Fine. And then we'll build the model, we'll train the model, we'll work with real world biomedical images. Okay. First, we'll use this architecture efficient net B0. Then we will be evaluating those results. Okay. We'll just not blindly believe the result. We'll evaluate. We'll use these things. And then at last, why 1 year access I have given you? We will be writing a manuscript together. If you wish to stick more than 6 months, then you can. You're welcome to stick. Okay. So yes, take a 5 to 10 minutes break, and I'll just send you another link. Roam around, just think what I've told you, just ponder over this problem, and then after a break, we'll just join again. Okay. Can I get a response?
Yes, ma'am. Okay.
Okay.
Okay,
Was it
ma'am. Thank you.
interesting? Was it interesting till now? Because we have not even started
Yes,
yet.
yes, ma'am.
Okay, okay. You will understand everything. Don't worry, okay? When we go for coding section also, you will understand. You don't need to memorize how to code. I'll tell you how you can learn. If you're not a coder, you can learn it. You're just a beginner. Okay, so yeah, I'm just stop sharing. Fine. So we'll meet within 5 to 10 minutes. I'll just check your mail also for the new link, okay? Start. Okay. So you type www.kaggle.com. Okay, you see, just like you create. I'm logged in. You won't be logged in. Just you create your you have created your Instagram. You have created your Facebook and all that, right? You have created your LinkedIn like that. Just create a profile over here, and okay, here you have to search. So the dataset name for our breast cancer is break his. Okay, dataset. So you see over here, so many break his dataset you are getting. And when we are working, I'll tell you which dataset I am working on. So if you look at Kaggle, here on the left hand side, you have got options like home, competition, benchmark, data hub, more, discussion. Okay, so like this, you can see. And here in your main page, welcome, your name, and here you see data set. So I have created 6 data sets over here. So you can see over here this 2 checkers. The first one is break his version 1 curated data set, because it's a very huge data set. You cannot load this data set directly in your laptop. Okay, so this was the raw data set. I curated it later on. So after curation, like what do you mean by cure? You cured it, right? You removed unwanted things which you don't want. I did that. And now this data set over here is only 4 GB. So if I open, so this, if you can see, you have 1, 2, 3, 4 folders, 100x, 200x, 400x, and 40x. So it contains images. Under 100x you have some benign cancer and benign tumor and malignant cancer. So AFPTTA, this is adenosis, A for adenosis and all those things. So can you see the tissue images, histopathological images over here? So see, not only CSV
Yes.
file, you can upload everything, whatever is necessary, you can upload anything. So you'll download the dataset from here, and then you also, the second dataset is UBC ovarian cancer. UBC, sorry, ovarian cancer. This one. So I have not, okay, this one I used from a competition in Kaggle. So Kaggle, you can, it hosts some competitions also. So you can participate in that also. So this is the data description. Right, you can download this. I'll just download it for you. You see you have 1057 files, and can you see the size? 794.01 GB. So you see how slow it might be. if you work in your laptop. And you can come to home, you can press create, you can create your own notebook. When you create, you click here dataset, you can upload your own dataset. There are some competitions that you also can create, so all these things are there. So it's very interesting things I learned from Kaggle. Apart from convolutional neural network, I had also learned graphical neural network just by participating in this particular Stanford RNA 3D folding competition. Although my rank was very bad, I came in some thousands of rank, but yeah, I had some, you know, I learned what is graphical neural networks, graph neural networks. So these are my submissions, I guess. No, not mine. Mm. My work is here. These are some things we have to do. So always start with EDA. EDA is exploratory data analysis. You will explore your data. Exploratory data analysis. And this I will tell you in phase 2 of the project. After the first 10 days, only then EDA. EDA, Joydeep, if you had remembered, okay, in our previous classes, I had taught about EDA also. You have to explore your data. And how will you explore your data? By coding. By coding only. You will first load your data, and then by the help of coding, you will understand how it is distributed. Whether it is a normal distribution, what is the mean, median, mode of these things, whether it is a balanced data or unbalanced data. Only after EDA, before proceeding to coding, you must understand your data, otherwise you cannot code properly. I guess it's enough for today. And so only these things were left, that this project, why does it matter? Okay, so this project, what it will do is, there's a biology. What is the biology? That you will see morphology, or how that tissue looks. Okay, and you will convert them into numbers. Okay, and then computer understand nothing but zeros and ones. It understand numbers. For making it more simple, what we do, we work with whole numbers, or we work with decimal numbers. You can note it down also if you are very new. So in computer terminology, decimals are known as floating numbers. Okay, it's float. So decimals whenever you understand, you come with the terminology float, it means it is numbers with points. Okay, I hope you understood. So if we work with this numbers, it is easier for computer to convert it into strings of zeros and ones into binary numbers, and then computer can learn. Apart, who had mathematics in +1, +2 level? Anyone? Anyone here had mathematics? Yes, no?
Yes, ma'am.
Harsha, you had maths. So you know about matrices, right? Matrices and determinants were a chapter.
Yes, ma'am.
Yeah. So our images will be converted into matrices only. So what do we have? Image, it's a colorful image, so it will have some channels: red, green, blue. We know RGB. Everyone, we know that there's RGB, right? RGB color, red, green, blue, and the colorful image is made up of RGB only. So how much red it is? It will assign certain number. 5, 1, 3. Sorry. Okay, 5, 1. Okay, I don't know what's happening. Yeah, this is the problem. Okay. So this is matrix. Matrix is nothing but a net of numbers, like a net. Actually, never call this a net of numbers, but people who don't understand maths, it will be like rows and columns, and each cell in Excel sheet, no, each cell they will have some values. So that will be converting into matrices. All images will be converted into matrices. So that's how we are converting the biology into computer ready data, and then we'll be processing it. So this project is not only a learning exercise. You will think and work like a computational biologist after this project. Okay, because we at the end of the day, we are solving a real world program, a real world problem. Okay, so what artifacts you will have? A coding notebook. A notebook is nothing but that file, that coding file that you'll have. You'll have beautiful figures and graphs. You'll have the final result. Your final result will be model, and maybe we can also deploy the model so that it will work either as a software or as a web based application. You can do that. Okay, if you do that, it's very, I won't say it is easy to publish, because if you're aiming for a good journal, it takes time for that. So yeah, only this portion was left. That's why I mm, just so I dragged this. The other classes won't be so slow. Don't think that the classes will be this boring from next time. No, because day one very less we had to cover. That's why it was. I talked with you people. I spoke to you. I told you some stories from my life. That's it. So next day, what we are learning. So you can go to the website. Nothing, you don't need to download for now anything. Everything is the first 10 days it will be available. If some links are not clickable, that means it will be updated as our class progresses. So second day we'll be understanding these histopathological images. Understood? So this is the diagram illustration. This is the real diagram. So not diagram, this is the real microscopic images. And I'll tell you one thing, this image, the normal breast tissue and the invasive ductal carcinoma tissue, this image is generated with the help of AI only. Just for the sake of this website, because I cannot upload, those images. Maybe if I have some issues, no, that's why I just made it from AI only. So, but you can see now how the pattern is different when it is a tumor pattern. It's so irregular, right? You can see these things. So we will understand what is tumor. We'll understand the biology first before jumping into coding. Please have patience. Let us understand the biology first, and then when we'll understand, only then again and again I'm telling you, we'll be able to make the computer understand that what work it has to do. So yeah, you can navigate very easily from my website, and you can go through the work. This is the day 3. And day 3 will be going through, okay, day 3 will be going through convolutional neural networks. Okay, and here we'll understand. You see the matrix, Harsha, I guess he knows the math. You can see the matrix over here, yes or no? Yeah. And you see how the matrix is generated in code like this. So you don't need to go much deep. You don't need to do that. And these are filter 1, filter. These filters are nothing but different layers. Yeah. And these are RGB channels. RGB, yeah, that means color channels. And you see this is the matrix. This is a 4 by 4 matrix. What do you mean by 4 by 4 matrix? That means you have 4 columns and you have 4 rows. And then we'll condense those 4 by 4 matrix. So how we are doing this? Yeah, this is the day 3 thing. Yeah. So if you feel that today's class is boring, slow, don't worry. We will pick up pace, and I hope you will understand everything. If not, I will go slow for every one of you. Don't worry. You can also mail me, message me on LinkedIn or WhatsApp, that which particular topic, or while we are starting to code, no, at that time you can tell me, ma'am, I did not understand this, please let me understand. Personally also I'll attend to your doubts. Okay, so that's it for today. And first let me close the recording, stop it.